The oxidative aerobic system. The oxidative system is the primary source of ATP at rest and during low-intensity activities and uses primarily carbohydrates and fats as substrates. Protein is normally not metabolized significantly except during long-term starvation and long bouts over 90 minutes of exercise. Approximately 70% of the ATP produced is derived from fats and 30% from carbohydrates. Following the onset of activity, as the intensity of the exercise increases, there is a shift in substrate preference from fats to carbohydrates. During high-intensity aerobic exercise, almost 100% of the energy is derived from carbohydrates. If an adequate supply is available, however, during prolonged submaximal steady-state work, there is a gradual shift from carbohydrates back to fats and proteins as energy substrates. Glucose and glycogen oxidation. The oxidative metabolism of blood glucose and muscle glycogen begins with glycolysis. If oxygen is present in sufficient quantities, the end product is glycolysis. Pyruvate is not converted to lactic acid but transported to the mitochondria, where it is taken up and enters the Krebs cycle, or citric acid cycle. The Krebs cycle is a series of reactions that continues the oxidation of the substrate. Begun in glycolysis and produces 2 ATPs directly from guanine triphosphate, GTP, for each molecule of glucose. Also produced from 1 molecule of glucose are 6 molecules of reduced nicotinamide adenine dinucleotide, NADH. Nicotinamide adenine dinucleotide, NDH, and 2 molecules of reduced flavin adenine dinucleotide, FADH2. These molecules transported hydrogen atoms to the electron transport chain, ETC, to be used to produce ATP and ADP. The ETC uses NADH and FADH molecules to rephosphorylate ADP and ATP. The hydrogen atoms are passed down the chain, a series of electron carriers known as cytochromes, to form a proton concentration gradient to provide energy for ATP production, with oxygen serving as the final electron receptor, resulting in the formation of water. Because NADH and FADH enter the electron transport chain at different sites, they differ in their ability to produce ATP. 1 molecule of NADH can provide 3 molecules of ATP, whereas 1 molecule of FADH can produce only 2 molecules of ATP. The production of ATP during this process is referred to as oxidative phosphorylation. The oxidative system, beginning with glycolysis, results in the production of approximately 38 ATPs from the degradation of 1 glucose molecule. 5.1 for a summary of these procedures. Fat oxidation. Fats can also be used by the oxidative energy system. Triglycerides stored in fat cells can be broken down by an enzyme, hormone-sensitive lipase. This releases free fatty acids from the fat cells into the blood, where they can circulate and enter muscle fibers. Additionally, limited quantities of triglycerides are stored within the muscle, along with a form of hormone-sensitive lipase to produce the intramuscular source of free fatty acids. Free fatty acids enter the mitochondria, where they undergo beta oxidation, a series of reactions in which the free fatty acids are broken down, resulting in the formation of acetyl-CoA and hydrogen atoms. The acetyl-CoA enters the Krebs cycle directly, and the hydrogen atoms are carried by NADH and FADH. Nicotinamide adenine dinucleotide and flavin adenine dinucleotide to the electron transport chain. An example of ATP production from a typical triglyceride molecule is shown in Table 5.2. Protein oxidation. Although not a significant source of energy for most activities, protein can be broken down into the major amino acids that are oxidized in the skeletal muscle are believed to be branched amino acids: leucine, isoleucine, and valine. Although alanine, aspartate, and glutamate may also be used. The nitrogenous waste products of amino acid degradation are eliminated through the formation of urea and small amounts of ammonia. The elimination of ammonia is significant because it is toxic and associated with fatigue. Control of the oxidative aerobic system. The rate-limiting step in the Krebs cycle, 5.6, is the conversion of isocitrate to alpha-ketoglutarate, a reaction catalyzed by the enzyme isocitrate dehydrogenase. Isocitrate dehydrogenase is stimulated by ADP, and normally inhibited by ATP. The reactions that produce NADH and FADH also influence the regulation of the Krebs cycle. If NAD plus and FAD plus are not available in sufficient quantities to accept hydrogen, the rate of the Krebs cycle is reduced. Also, when GTP accumulates, the concentration of succinyl-CoA increases, which inhibits the initial reaction oxaloacetate and acetyl-CoA to citrate and CoA of the Krebs cycle. The electron transport chain is inhibited by ATP and stimulated by ADP. The simplified overview of the metabolism of fat, carbohydrate, and protein is on 5.7. Energy production and capacity. The phosphagen, glycolytic, and oxidative energy systems differ in their ability to supply energy for activities of various intensities and durations. Exercise intensity is defined as a level of muscular activity that can quantify into terms of power, work performed per unit of time output. It is shown that power at maximal oxygen uptake is approximately 20 to 30% peak power on a cycle ergometer. Therefore, exercise primarily supported by aerobic mechanisms, even at 100% of maximal oxygen uptake, should not be classified as high-intensity exercise. Activities such as resistance training that are high intensity and thus have a higher power output require a rapid rate of energy supplied. They rely almost entirely on the energy supplied by the phosphagen system. Activities that are low intensity but long duration, such as marathon running, require large energy supply. They rely on energy supplied by the oxidative energy system. The primary source of energy for activities between these two extremes shifts depending on the intensity and duration of the events, table 5.3. In general, short, high-intensity activities, resistance training and sprinting, rely on the phosphagen energy system and fast glycolysis. As the intensity decreases and the duration increases, the emphasis gradually shifts to slow glycolysis and the oxidative energy system. The duration of the activity also influences which energy system is used. Athletic events range in duration from 1 to 3 seconds, snatch and shot put, to more than 4 hours, triathlons and ultra marathons. If an athlete makes the best effort, an effort that results in the best possible performance for a given event, the time considerations are shown in 5.4 are reasonable. At no time during either exercise or rest does any single energy system provide the complete supply of energy. During exercise, the degree of which anaerobic and oxidative systems contribute to the energy being produced is determined primarily by the exercise intensity and secondly by the exercise duration. Substrate depletion and repletion. Energy substrates, molecules that provide starting materials for bioenergetic reactions including phosphagen, ATP, and creatine phosphate, glucose, glycogen, lactate, free fatty acids, and amino acids can be selectively depleted during the performance of activities of various intensities and durations. Subsequently, the energy that can be produced by the bioenergetic system is reduced. Fatigue experienced during many activities is frequently associated with the depletion of phosphagens and glycogen. The depletion of substrates such as free fatty acids, lactate, and amino acids typically does not occur to the extent that performance is limited. Consequently, the depletion and repletion patterns of phosphagens and glycogen following the physical activity is important in exercise and sport bioenergetics.